Good evening, everyone. Myself, Dr. Aanchal, and this is a new series that we have launched. That is 45 days to MBBS finals intensive revision series, and in which in this series we're going to do rapid revision of all the topics, volatile facts through quick revision. We discuss here. High topics will be covered, and the most expected topic will cover in this series, which are beneficial for everyone and also for all the university exams, right? So in today's class, we're going to have the revision over the blood, and in blood we're going to do the RBC destruction, hemoglobin, anemia, and blood indices. So we're going to have two short essay question and two short answers. In short essay questions, we're going to know about the hemoglobin synthesis, and we're going to study about the hemoglobinopathies. Then we're going to discuss about the fate of hemoglobin. Then in short answers, we're going to discuss about the different types of hemoglobin, fetal hemoglobin, functions of hemoglobin, and also in the end, we're going to understand the MCH and MCV, and we're going to have a MCQ over both of these. Then in Auricle, we provide live classes for the elevated learning of the students. Then also recorded sessions are there. So that the student will have the access of all the subjects anytime, anywhere through the recorded sessions. Then also we provide exam modules so that the student can cover the entire syllabus in some particular hours. Then also we provide practical sessions. Practical sessions is for the learning of the student so that the practical part will be covered properly. And also we provide one-to-one doubt clearing session so that the student have any kind of doubt. So the student can clear through this doubt clearing sessions. And a question bank is there so that the student can have the different types of MCQs, and MCQs are here from beginner level to expert level. Then hard copy notes also we provide for the students. Then in Auricle, we have subscription plan starting from basic to mentorship plan. Basic plan is of 3 months, standard plan is of 6 months, and mentorship plan is of 12 months. In mentorship plan, we provide all the services that is provided by Auricle, like pre-recorded classes, live class, slide out clarification, one-to-one personalized guidance, notes, quick revision examination module, and this plan costs around 50,000. Also for instant 20% discount, you can use my code, that is Dr.Aanchal 20. So let's start with today's class. So first question we have to describe the steps in hemoglobin synthesis and also to write a note on the disorder of hemoglobin synthesis and mention two types of hemoglobinopathies. So first we're going to discuss about the hemoglobin synthesis, right? So synthesis of hemoglobin, we know that the synthesis of hemoglobin starts in which stage of erythropoiesis? It starts in pro-erythroblastic stage, right? Already we have studied this. So we know that the hemoglobin start appearing in which stage? Hemoglobin start appearing in intermediate normoblast. And production of hemoglobin is continued until the stage of reticulocyte, and finally after reticulocyte we get the mature RBC, right? So heme portion of hemoglobin is synthesis in mitochondria, and the protein part, globin, gets synthesis in hemoglobin. Okay, again I'm repeating that heme portion gets synthesized in mitochondria, and we get the globin in ribosomes. Okay, so as we can see in the slide also, these are the various steps that we're going to understand one by one in detail with the help of the diagram. So in the next slide, I'm going to draw our mitochondria, in which we're going to know that how the synthesis of heme take place in mitochondria. So this is, we have mitochondria, and here we have cytoplasm. Okay, so with the help of this diagram, we're going to understand that how heme synthesis take place. Okay, so heme synthesis take place from succinyl-CoA. As we can see, the succinyl-CoA plus glycine together constitute the heme synthesis, right? So succinyl-CoA and glycine together form the heme. Okay, so the sequence of events are there. So first we have that first step in heme synthesis take place inside the mitochondria, as already we have drawn. So here two molecule of succinyl-CoA and two molecule of glycine together condense to form a product that is known as delta amino leuvinilic acid. Okay, and this occurs with the help of an enzyme that is known as ALA synthetase. Okay, amino leuvinilic acid synthetase. Okay, now what happened? This is the first step that we have learned, that two molecules of succinyl-CoA and two glycine together constitute delta amino leuvinilic acid. Okay, now what happened? This product that we have constituted will go into the cytoplasm. Okay, will go into the cytoplasm, and ALA is transported to the cytoplasm, and two molecules here. So two molecules will going to combine and so two molecules of this product ALA combine to form another product that is known as porphobilinogen. Okay, and this product formed in the presence of an enzyme known as ALA dehydratase. Okay, so this is how it take place. Porphobilinogen take place in the presence of an ALA dehydrogenase, right? Now, what will happen next? Now, this porphobilinogen will start constituting, or it get converting into another product that is known as uroporphobilinogen. Okay, by an enzyme known as here, by an enzyme known as synthase. Okay, now what will happen as this uroporphobilinogen synthesis take place? That is basically the first one. Okay, so this is known as uroporphobilinogen 1. Now this uroporphobilinogen 1 is converted into uroporphobilinogen 2. And this uroporphobilinogen 2 get converted into uroporphobilinogen 3, right? Now, from this third product, finally, we constitute a product that is very essential. So, from this uroporphobilinogen 3, we get a final product that is known as porphyrinogen 3. Okay, now what will happen? This porphyrinogen 3 is transported back to the mitochondria. Okay, so here this porphyrinogen 3 transported back to the mitochondria, and here it get oxidized. So the oxidation will take place, and finally it will constitute protoporphyrinogen 9. Okay, so this steps takes place by oxidation of chlorophyrinogen 3. Okay, now finally this protoporphyrogen get converted into protoporphyrin 11, right? Now what will happen? This protopyrinogen 11 is converted into protopyrin 9. Okay. Now 9 combines with iron to form heme in the presence of an enzyme that is known as ferrochelatase. Okay, so this is how the synthesis of heme take place. Okay, so finally the synthesis of heme take place inside the mitochondria. Okay, now next we have the formation of globin. Okay, so polypeptide chains of globin are produced in the ribosomes. Okay, so you have to know that ribosomes are responsible for the production of globin. So there are 4 types of polypeptide chains, namely alpha, beta, gamma, and delta. And each of these chains are different from each other by amino acid sequence. Means in each chain, different amino acid sequence are present. So here in adult hemoglobin, 2 alpha and 2 beta chains are there. And in fetal hemoglobin, 2 alpha and 2 gamma chains are there. So the different synthesis of different types of chain is due to the different sequence of amino acids that is present in globin. Okay. Fine. So this is how we have covered the first step, that is the hemoglobin synthesis. Now next we have the disorders of hemoglobin synthesis, and we have to know about the hemoglobinopathies, right? So in this, we're going to know about the different abnormal hemoglobins. Okay, so we have abnormal hemoglobin. Then we're going to know about the hemoglobinopathy, cause, and finally changes in hemoglobin structure. Okay, so first we have the hemoglobin S, the most common form that we get. So hemoglobin S, the hemoglobinopathy we have is due to sickle cell anemia. Okay, and we know that in normal chain, if we talk about, so normal chain is alpha and beta, but here there will be substitution of amino acid valine for glutamic acid at 6th position of beta chain. Okay, so we can write that here substitution will occur of amino acid valine for glutamic acid at 6th position of beta chain. Fine. Then next we have hemoglobin C. So here we have the hemoglobinopathy causes hemoglobin C disease. So here we know that in normal hemoglobin we have alpha and beta chain, but here we have substitution of amino acid lysine for glutamic acid at 6th position of beta chain. Okay, so again there will be substitution of amino acid lysine for glutamic acid at sixth position of beta chain. Fine. Then third we have hemoglobin H, and this hemoglobin H is commonly known as a type of alpha thalassemia. Okay, so what happened in alpha thalassemia? The A chain get decreased or absent, or it will be abnormally synthesized or abnormally present. Okay, so alpha chain is decreased, absent, or abnormal. Okay, so here we have covered the different type of hemoglobinopathy, the most common type. Okay, fine. Now next we have, so another question we have is the fate of hemoglobin, the damage to red blood cells, and the following the hemolysis. Okay, so now we're going to discuss about the fate of hemoglobin. Okay, so whenever we talk about hemoglobin, so we know that RBC has a lifespan of 120 days, and after the lifespan of 120 days, what will happen? This will lead to destruction of RBC, right? So after the lifespan of 120 days, the destruction of RBC will occur, and that destruction will occur in reticuloendothelium system, right? So particularly in spleen. So what will happen as the RBC get destroyed, there will be ejection of hemoglobin. Okay, now the question is that what happened to this hemoglobin? So what will happen to this hemoglobin, that we're going to understand with the fate of RBC and in the fate of hemoglobin. Okay, fine. So as we can see in the picture also, RBC is there. When the lifespan of RBC got end, means 120 days get completed. So that gets stored inside spleen, right? So now hemoglobin get ejected out, and by the phagocyte, like macrophages are there. So it get converted into iron plus porphyrin portion. Okay, so that porphyrin portion get converted into biliverdin, then bilirubin, then reaches to blood for the conjugation, and finally the conjugated blood bilirubin get converted either into stercobilinogen or urobilinogen. Urobilinogen circulated in blood, and stercobilinogen eliminated in the feces. Okay, so this part of hemoglobin, fate of hemoglobin, we're going to understand again with the help of a flow chart. Okay. So as we have discussed that as the RBC lifespan over. Now the hemoglobin ejected. Right? Now what will happen to this hemoglobin? This hemoglobin can split into two parts, heme part and globin part. Okay, so there we have heme part, then we have globin part, right? So this globin part enters into amino acid pool cycle. Okay, and again it will be reutilized for the synthesis of hemoglobin. Now what will happen to this heme part? Okay, this heme part is the iron protoporphyrin 9. Okay, so it gets split into two parts. The first we have iron, and the another is the remaining part. Okay, so this iron, that is iron Fe2 plus, get combines with apoferritin. Okay, and it will become ferritin and stored inside liver, so that it can be reutilized again. Okay, so it will be reutilized for the synthesis of the hemoglobin. Okay, Now, what will happen to this remaining part? So, this remaining part first get oxidized by the enzyme known as hemoxygenase. The name of the enzyme is very important. It's usually asked in the MCQs also. So, first it get converted into biliverdin. Then, next, there will be reduction, and that reduction will occur with the help of biliverdin reductase, and finally we get the bilirubin. Okay, so bilirubin is the product that get excreted into bile. Okay, so this is all about the fate of hemoglobin, the fate of RBC. Fine. Then next we have the type of hemoglobin. Okay, so there are variety of hemoglobin that are present. So type of hemoglobin is totally depend upon the composition of chain. Okay, so as we can see, we have HbA. HbA is hemoglobin adult. Then HbA2, that is hemoglobin adult 2. Okay, so two forms of hemoglobins are present in adult hemoglobin, HbA1 and HbA2. Now, what is the difference between HbA1 and HbA2? In HbA1, 2 alpha and 2 beta chains are present, whereas in HbA2, 2 alpha and 2 delta chains are present. Okay, but majorly the main concentration that is present inside adult hemoglobin is HbA1, that is approximately 90%, whereas in HbA2, this HbA2 is only present approximately 5% less or more than now 5%. Okay. Then another we have fetal hemoglobin and HbA1c. Okay. So first we're going to talk about fetal hemoglobin. So fetal hemoglobin, we have alpha 2 and gamma 2, that is approximately 2%, less than 2%. Then we have HbA1c. Then in that we have alpha 2, beta 2, glucose chains, and approximately it is also the 5%. Okay, so these are the different types of hemoglobin that is present, and the main difference in all the types of hemoglobin is the composition of chain. And this question is important that because it has already asked in the previous year question also. So all the students should remember these things. Okay. The next we have the fetal hemoglobin. Okay. This question is also important because it is also already asked in various university exams. So we're going to understand the fetal hemoglobin. So already we have talked about fetal hemoglobin, that in fetal hemoglobin, the alpha 2 and gamma 2 chains are present. Okay. So its structure is same as that of hemoglobin adult, HbA, except that beta chains are placed by gamma chains. Okay. And here gamma chains also contain 146 amino acids, but have 37 amino acids that is different from beta amino acid. Okay. So particularly if we talk about the main difference between HbA and HbF is the amino acid. The sequence of amino acid is different. Okay. Then another thing that this fetal hemoglobin is much more resistant to the action of alkalis than than adult hemoglobin. Okay. So another we have that this fetal hemoglobin is more resistant to the action of alkalis. Okay. So first we have 2 alpha and 2 gamma chains are present. Okay. Then this fetal hemoglobin has greater affinity for oxygen. Okay. So as we can see in the diagram also, if the normal affinity for adult hemoglobin is this, but the fetal hemoglobin curve is more shifted towards left, means the affinity for oxygen is more in fetal hemoglobin than in adult hemoglobin. Okay. And why this occurs? Because of poor binding of 2,3-DPG. Okay. So another thing that it has poor binding of 2,3-DPG. G. So in another term, we can say that it can take much larger volume of oxygen than HbA at even lower pressure too. Okay. So this facilitates the movement of oxygen from maternal to fetal circulation, and HbF is saturated at 20 mmHg of PO2, whereas the HbA is only 30 to 35% saturated at this pressure. Okay, so all the student, please remember that the HbF has greater affinity for oxygen as compared to adult hemoglobin. So it can take more or much larger volume of oxygen than that of HbA. Okay. Then coming to the lifespan. So we know the lifespan of RBC, the adult RBC is 120 days. But here the lifespan of fetal RBCs are very less. So it is approximately 80 days. Okay. Fine. So fetal hemoglobin has higher affinity for PO2 for O2 at all partial pressure. This ensures that O2 is transferred to the fetus from maternal blood across the placenta. Then the PO2 in fetal tissue is very low due to high metabolic rate associated with fetal growth rates. Then although fetal 2B has a higher affinity for oxygen in such a low partial pressure environment of the fetal tissue. It unloads oxygen readily, and at birth, the fetal hemoglobin is replaced by adult hemoglobin. Okay, so actually, what happened as the birth of the fetus will take place? So after 2 to 3 days of the birth, there will be change in the fetal hemoglobin. The fetal hemoglobin start replacing by the adult hemoglobin. Okay. Now, next we have mentioned the function of hemoglobin. So the function of hemoglobin is very, very important, as already it has asked in the various previous year questions also. So please remember all the function. Okay. So we know the main function of hemoglobin is the respiratory mechanism. Oxygen is transported and it is very important for the ATP generation. Okay. Then another functions are also there. So one by one, we're going to discuss about the functions of hemoglobin. So first function we have that it facilitates the transport of O2 from lungs to tissues. Okay. So as we inhale the oxygen, so the oxygen will be taken to the lungs, into the alveoli. It can diffuse across the blood capillaries, and blood capillaries will transport the oxygen along with the hemoglobin to the tissues. Okay, so this is the main function. Then another function that it also facilitates the transport of CO2 from tissues to lungs. Okay. So as we know, inside the body, various metabolic reactions take place. Okay. So by those metabolic reactions, there will be generation of CO2. So that CO2 will taken by the blood vessels again to the lungs, and with the help of exhalation, we actually exhale the CO2 from out of the body. Okay. Then another we have acid-base buffer. It also acts as a acid-base buffer. Okay. So being a protein, it acts as an excellent acid-base buffer, and it is responsible for 70% of buffering power. Okay. Then next we have the hemoglobin bind with nitric oxide in the lungs, and it release into the tissue where it dilates the vessel. Okay. So the HB binds with nitric oxide in lungs and releases in tissues where it helps in the dilation of blood vessels. Okay, so these are the main function of hemoglobin. Okay, now next we have the question over the MCV and MCH. So there is a question that we have to solve. Okay, so first we're going to understand that what is MCH and MCV. Okay, so first we're going to see here the question we have: calculate MCV and MCH. Given is PCV, that is 45%. RBC count we have is 5 million, and HB we have is 15 gram. Okay, so with the help of PCV, RBC, and hemoglobin concentration, we have to calculate the MCV and MCH. So for MCV, we have a formula that is PCV percentage into 10 upon RBC count. Okay. And for MCH, we have another formula that is hemoglobin upon RBC count into 10. Okay. So what was the formula for MCV? That was PCV upon RBC into 10. Okay. And what was given in the question? The given was PCV, that is 45%, RBC count, that is 5 million, and hemoglobin, that is 15 gram. Right? Now, one by one, with the help of this formula, we're going to calculate. First, we're going to know about the MCH. So, we have 15 into 10 in upon 5. Okay. So, we'll have 30. Okay. So, that is basically picograms. Okay. Then, next we have 45 into 10 upon 5. So we get 90, right? And here we have in femtoliters. Okay, so with the help of the formula of MCH and MCV, and with the help of these value, we are able to calculate the MCV and MCH. Okay. So this is all for today's class. I think we have covered all the questions, right? Let's have a look again. So we have covered the hemoglobin synthesis, all the steps we have seen, then fate of hemoglobin we have done, types of hemoglobin we have done, fetal hemoglobin is done, then function of hemoglobin we have seen, then also in the last we have calculated MCV and MCH. Okay. So this is all for today's class. I hope you find this class more helpful for you. So thank you so much for watching. And for any type of query and questions, you can connect to us through various social media platforms like YouTube, Instagram, Facebook, and Telegram. So thank you so much for watching my class.